from Group 6 will be presenting to you how nutrigenomics can cause hypertension. This is our group members. Do you know what is nutrigenomics? Nutrigenomics is the fusion of nutrition and genomics. Nutrition, as you likely know, deals with the stuff you eat. Its nourishment and the field of nutrition looks more closely at the component of what you are eating. It all relates back to food. Meanwhile, genomics is related to genetic, the study of the complete set of DNA in a person or other organism. Essentially, genes makes who you are. They determine your traits and to what extent your body responds to various stimuli. Thus, it's the study of consequences of the influence nutrition and other bioactive food component on the expression of the genetic material. Example, excessive intake of salt and saturated fat can alter our gene expression and causes hypertension. What about nutrigenetics? Nutrigenetics identify how the genetic makeup of a particular individual coordinates their response to various natural response. It also reveals why and how people respond differently to the same nutrient. Have you ever wondered how nutritional genomics in this era has evolved? There are many things that have been improved, but today I will focus on personal nutrition. In the early 1920s, Dr. Thomas starts using dietary therapies to treat Bright's disease, recognizing the needs to individualize dietary therapy for optimal result. In early 2000, human genome decoding leads to the identification of genetic variants or a single nucleotide polymerism related to individual sensitivities to lifestyle and diet. And currently, the study of nutrigenomics has taken off using data from the non-coding areas of the human genome with genomics and epigenomic trends. Personalized recommendation based on genetic and biometric replace one-size-fits-all advice, revolutionizing healthcare and medical nutrition therapy.
Hi, my name is Nuresha binti Muhammad Zaidi with message number 068040, and I will continue the presentation with potential role of nutrigenomics to dietitian or nutritionist. The first role of nutrigenomics is nutritional optimization. Dietitian or nutritionist can use nutrigenomics to help them determine an individual ideal nutrient intake based on their genetic makeup. According to the client data, dietitian can identify genetic variation that impact nutrient absorption by using single nucleotide polymorphism. For example, if an SNP is identified within a gene encoding a key enzyme for nutrient absorption, and it is known that this SNP results in less effective absorption, thus a higher intake of that nutrient would be recommended for the client. The second potential role is dietitian and nutritionist can provide nutrigenomic counseling and guidance on how diets affect the gene's expression. This counseling aims to match clients to the food that's suitable for them. The last potential role is disease prevention and management. Nutrigenomics can provide valuable information on genetic predisposition to chronic disease such as obesity, diabetes, cardiovascular disease, and certain type of cancer. By understanding an individual genetic susceptibility, dietitian or nutritionist can develop target strategies to prevent or manage this condition through personalized nutrition intervention. They can focus on specific nutrients, dietary patterns, or lifestyle modification that can offset genetic risk and treat the illness.
Next, let's talk about the benefits and misleading claims in nutrigenomics. There are several benefits about nutrigenomics. Firstly, by using nutrigenomics, we can upgrade our skills and knowledge about the relationship between the nutrients and our genes. Next, we also can learn about the unique of genes of each patient. For example, patient A with diabetes mellitus might have different effect by consuming brown sugar compared to the other patient. Other than that, we also can see the proof of nutrigenomics itself. We surely understand how the nutrients can affect our genes. Next, by understanding the nutrigenomics, we can improve our fitness and health level by practicing the good effect of nutrigenomics. For instance, a person with insulin resistance genes can have a better health by having a good diet habit. Now, let's turn to the misleading claims about nutrigenomics. Firstly, from past research, we actually do not have a solid proof that altering our diet might eliminate the bad genes. Next, we can't advise the patient to alter the diet if they do not have any evidence or medical checkup from the trusted medical experts. It might lead to bad effects of the patient or even can lead to death. Furthermore, nutrigenomic misleadings has always been related to disease that actually not fully diagnosed and confirmed yet. For instance, people with cancer gene might not necessarily having cancer in their later life. Lastly, tests about disease that related with genes are not always reliable to us.
Assalamualaikum. My name is Nur Husna. I'm going to continue on explanation potential role of nutrigenomics in managing the hypertension and preventing hypertension. By knowing genetic variants can help managing hypertension because there are genetic variants influence process and respond to the nutrients. Genetic analysis of Mendelian has highlighted that finding comprise a rare genetic variants with large impact on blood pressure. Next, about the sodium sensitivity. Nutrigenomics have identified genetic variants associated with the sodium sensitivity. There are evidence that genetic predisposition modulate the blood pressure response to the diet. So, dietitian can tailor suggestions to restrict sodium consumption by studying the person's genetic propensity. Next is preventing hypertension. In preventing hypertension, genetic risk assessment may help because it can identify genetic variants associated with increased risk of developing hypertension. So, we can take a proactive step to reduce the risk of hypertension. Next, by personalized dietary recommendations. Based on person genetic profile, Nutrigenomics can personalize dietary advice to reduce the hypertension. For example, recommendation on the specific dietary patterns like a DASH diet but according to the food that person can be consumed.
Now, let's look at two genes associated with hypertension. They are angiotensin converting enzyme gene and angiotensinogen gene. Both genes play a role in the renin-angiotensin-aldosterone system, which regulates blood pressure and fluid balance in the body. As for AGT gene, it encodes angiotensinogen, a precursor protein that is converted to angiotensin 1. Angiotensinogen is produced by the liver and released into the bloodstream. Variation in the AGT genes, such as the M235T polymorphism, has been investigated in relation to hypertension. The T allele has been associated with higher level of angiotensinogen and an increased risk of hypertension. The ACE gene encodes the angiotensin converting enzyme, which converts angiotensin 1 to angiotensin 2. Angiotensin 2 is a potent vasoconstrictor that causes blood vessels to narrow, leading to increased blood pressure. Variation in the ACE gene, such as the insertion or deletion I or D polymorphism, has been studied in relation to hypertension. The presence of D allele has been associated with higher level of ACE activity and increased risk of hypertension.
These are the conclusions based on study findings on the ACE and AGT genes. ACE gene encoded for ACE enzyme is crucial in RAS by converting angiotensin 1 into the strong vasoconstrictor angiotensin 2, which is also important for controlling blood pressure. One of the variations is intron 16 of the ACE gene's non-coding region insertion or deletion polymorphism, which can affect the existence or absence of the 2X7 base pair DNA sequence. The current study also showed that the Asian, Caucasian, and misgender populations were more susceptible to hypertension and mostly connected with the ACE gene D allele. One of RAAS essential enzyme is AGT because it contributes to the productions of angiotensin 1 and 2. It plays a part in controlling blood pressure. Pathogenesis of a few disorders is significantly influenced by AGT M235T and T174M. More diseases have been linked to these two variants of AGT. AGT M235T is observed to be related to hypertension and numerous cardiovascular illnesses. These two variants were found to be more prevalent than the other polymorphisms in various groups.
Hi, I am Shabrina. So now I would like to share about status Nutrigenomics acceptance in Malaysia. We look at the result of one survey which analysed by SPSS software, and the result shows positive acceptance towards Nutrigenomics that involved 421 respondents. Secondly, based on one article, first Nutrigenetics and Nutrigenomics Research and Training Unit was established in Malaysia on February 2022. Overall, based on these two evidence, Nutrigenomics is a relatively new field in Malaysia, but there is growing interest and for its acceptance in Malaysia. Next is hope for the success of Nutrigenomics practice in Malaysia. In order to succeed, government need to increase public awareness and acceptance in Nutrigenomics. Educating the public about the benefits of personalized nutrition and its potential impact on health outcome is essential. Initiatives that raise awareness and promote understanding can help pave the way for the success of Nutrigenomics in Malaysia. Besides, collaboration between researcher, healthcare provider and policymaker is crucial for the successful implementation of nutrigenomics, especially in hypertension management. Interdisciplinary collaboration can lead to a deeper understanding of the genetic and dietary factors and at the same time contributing to success of nutrigenomics practice in Malaysia. I think that's all from us. Thank you.